Welcome to General Biology. Today's presentation is going to be on Mendelian genetics. There are several new terms we have to cover when it comes to Mendelian genetics. Two of those terms are genes and alleles. So a chromosome is something we find genes on, and we get one chromosome from our mom and one chromosome from our dad. And the genes on those chromosomes aren't always the same that we get from mom and dad. And so you can see in my example here, we have two chromosomes, a mom chromosome and a dad chromosome. The genes on these chromosomes are gene A, B, and G. And the alleles are the forms of that gene that I get from each parent. So here I get a capital A from dad and a lowercase a from mom. Capital B from mom, lowercase b from dad. And in this case, in the G case, I got a capital from both parents. So whether it's capital or lowercase, that's the form of the allele. The letter is the gene, and the chromosomes are the structure you see. We also have the terms dominant and recessive. So a dominant gene is one that is expressed or shows up if it's present. So you can see in this diagram, anywhere we see a big B, you have brown eyes. In order to get a recessive trait to be expressed or show up, you have to have two copies of that gene. So in this particular case, in the blue eyes, you have to be little b to have blue eyes. Now, eye color is more complicated than this. There's several genes that are involved in eye color, but this is just a good picture to use as an example. Some more terms that we're going to need to know are genotype, phenotype, and homozygous and heterozygous. So a genotype is the actual genes that you have, gene type, and your phenotype is your appearance. So I think pheno-photo. So your phenotype is your appearance. So here we have this P has the genotype big S. This P has the genotype big S little s. Now, they both have the exact same appearance. So they would have the same phenotype, but a different genotype. And then these two P's have different phenotypes, round versus wrinkled. When we're looking at genotypes, they can either come in a situation where both of them are the same, and if that's the case, we call it homo. And if they're different, we call it hetero. So we have a homozygous situation here, a heterozygous situation here, and another homozygous situation here. When Mendel did all of his crosses, he would do several generations. This is just one example he did with pea color. And so he made it a yellow with a green pea, and all of the offspring were yellow. And then he mated two yellows together, and he got 3/4 yellow and 1/4 green. And so he came up with the idea of the 3-to-1 ratio showing up. And he ended up figuring out what he called the laws of independent assortment and the laws of segregation. And so he did this with several different characteristics in pea plants. He looked at flowers, whether or not they were purple or white, whether or not they were at the end of the plant, terminal, or along the edges of the plant, axial. He looked at pea color, whether it was yellow or green, pea shape, whether it was round or wrinkled, whether the pods were inflated or constricted, whether the pods were green or yellow, and whether or not the plants were tall or short. So he looked at all these traits, and he did lots and lots of crosses and came up with thousands and thousands of seeds, and he came to some conclusions. And two of his conclusions were that genes or alleles are going to separate when you make uh, your gametes, and that they're going to separate independently from each other when you make your gametes. So he came up with the law of segregation and the law of independent assortment from all of his experiments and his mathematical results.